We recently brought you the story of a submersible called Limiting Factor. It was used to reach the deepest parts of all 5 of the Earth's oceans. We took you along as it reached Challenger Deep.
Nearly 8,000 meters beneath the Atlantic Ocean, scientists made a discovery in the Puerto Rico Trench that defied all expectations.
Why do we need to go to the depths of the ocean in this way?
Well, the oceans constitute 70% of our planet, and of those oceans, 80% are still unexplored, so half of the world is still completely unexplored.
What they uncovered doesn't just challenge scientific theories, it completely upends our understanding of the planet. This isn't some fictional tale, it's a real and groundbreaking revelation. The implications are enormous, sparking intense curiosity and debate in the scientific community. Join us as we uncover the astonishing truth about what lies hidden deep below the surface of the trench. Evidence of mega-earthquakes and tsunamis. The Puerto Rico Trench signifies not just the deepest point in the Atlantic Ocean, but it also hides the geological scars left by massive seismic activities. In 2023, a collaborative effort between the USGS and the University of Puerto Rico led to the recovery of sediment cores from a mangrove pond located at East Bajura, in the northwest region of Puerto Rico. These cores revealed a notable sand layer, measuring half a meter in thickness, which exhibits the characteristics of tsunami deposition, an eroded base, graded sediment, and a chaotic mix of marine microfossils with terrestrial materials. Radiocarbon dating indicates that this deposit dates back to between 1470 and 1530 CE. Geological models suggest that such a deposit is the result of a megathrust earthquake, with a magnitude of 8 or greater occurring along the Puerto Rico Trench. Supporting evidence comes from tsunami deposits of a similar age found up to 400 kilometers east in the Virgin Islands and Anegada in the British Virgin Islands. In these areas, fields of coral-clast and mollusk boulders stretch over a kilometer inland, far exceeding the reach of hurricanes, and are primarily dated to between 1200 and 1480. Modeling indicates that a rupture length of 300 to 400 kilometers and magnitudes ranging from 8 to 9 would be necessary to create such significant overwash. These findings suggest that the subduction interface of the trench experiences periodic ruptures during megathrust events, presenting risks not only locally but throughout the Caribbean basin. Multibeam bathymetry, conducted north of Puerto Rico, has uncovered a multitude of retrogressive slope failures along the steep edge of the carbonate platform. The presence of large submarine slides, including a significant 7 times 3 kilometers slump head, suggests that slumping is an ongoing process that could potentially displace enormous volumes of water, leading to tsunami generation even in the absence of major seismic events. The NOAA Ocean Explorers surveys from 2003 have documented these submarine scarps and fissures, emphasizing the dual role of tectonic ruptures and mass-wasting processes in heightening tsunami risk in the area. Paleoseismic investigations utilizing sediment cores, CT imaging, and X-ray fluorescence have enhanced the understanding of the timeline of these occurrences. Detailed analyses of grain size distributions and microfossil assemblages found in coastal ponds north of Puerto Rico have revealed several sand layers that correspond to distinct tsunami events. The unique mineral signatures and basal scour surfaces of each layer serve to differentiate tsunami deposits from typical storm overwash, highlighting the trench's history of infrequent yet catastrophic megathrust earthquakes. The seismic and tsunami potential of the trench is crucial for hazard evaluation. Simulations of earthquakes that include the event from 1470 to 1530 CE indicate that tsunami waves might traverse the Atlantic, affecting remote coastlines. At the same time, the ongoing multi-beam mapping is uncovering new slope failures and fault structures that may lead to future ruptures. It is vital to combine geological records with geodetic and seismic monitoring to predict the next megaquake and equip communities across the Caribbean for its extensive impacts. What if the ocean's deepest fish turned out to be something else entirely? Hundreds of ribbony creatures once mistaken for fish now redefine Hadal life. That leads us to the next, holothurians, sea cucumbers. In 1964, Perez documented swarms of snailfish along a 1-mile transect at a depth of 7,300 meters, estimating approximately 200 individuals, an impressive density for such profound depths. No specimens were gathered, leading to the assumption for decades that these were Liparidae. However, when Victor Vescovo's DSV limiting factor returned in 2019, the cameras did not capture any snailfish. Instead, they recorded numerous elasipodid holothurians. These sea cucumbers, belonging to the genus Peniagone, are morphologically nearly indistinguishable in color, texture, and undulating movement. Their significant presence, hundreds per 100 meters, challenges the previous snailfish speculation and firmly establishes holothurians as the dominant megafauna of the trench. In addition to Peniagone, archival research and reevaluations of photographic evidence have identified at least 2 other holothurian types. Heather Stewart highlights 3 distinct morphotypes: a Synalactidae species, potentially Pseudostichopus or Mesothuria, observed around 25 times, measuring approximately 10 to 12 centimeters in length; a flatter, ribbon-shaped holothurian of similar dimensions, seen only a handful of times; 2 very long cylindrical specimens approximately 30 centimeters in length and 4 centimeters in diameter, gray in color, which leave deep sediment trails, tentatively classified under the genus Myriotrachus. These deposit feeders move slowly over the abyssal mud, utilizing their tube feet and tentacles to gather organic detritus. At a depth of 7,300 meters, biologists observed mounds measuring up to 40 centimeters in width and 25 centimeters in height, likely serving as feeding stations or burrow entrances for holothurians. The presence of spiral trails and sediment reworking highlights their ecological function, breaking down particulate matter and cycling nutrients in an environment where food is only occasionally supplied by surface productivity. The taxonomic resolution is still inadequate. No specimens from the trench floor have been formally collected for museum documentation. Consequently, all identifications are based on in situ morphology. Nevertheless, the remarkable density of Peniagone spearns, which reflects the original snailfish counts, emphasizes the trench's surprising benthic biomass. At pressures surpassing 700 bar, temperatures close to 2 degrees Celsius, and in complete darkness, these holothurians flourish in conditions where few metazoans can survive, showcasing extraordinary physiological and structural adaptations. Gravity anomaly and mega-thrust quakes. The Puerto Rico Trench stands as the deepest point in the Atlantic Ocean and is also home to the most significant negative free air gravity anomaly on the planet, measuring 380 million gallons of water. This anomaly is situated approximately 50 kilometers south of the trench axis at a depth of around 7,950 meters. The notable gravity low is indicative of an active downward force from the subducting North American slab, which bends and descends beneath the Caribbean plate. The oblique convergence, occurring at an angle of 10 to 20 degrees to the trench axis, results in both subduction and left lateral strike-slip motion. This process has tilted a previously horizontal late Oligocene carbonate platform northward by about 4 degrees in less than 40,000 years, with its northern edge now resting at approximately 4,000-meter depth, and its reconstructed landward edge at an elevation of plus 1,300 meters in Puerto Rico. Seismic and gravimetric analyses suggest a probable tear in the descending slab near the longitude of 64.5 west. This inference is supported by clusters of small magnitude earthquakes and significant variations in gravity and bathymetry along the strike. Notably, 2 earthquake swarms in 2007, occurring in April and July, exhibited trench parallel tensile stresses at depths of 50 to 100 kilometer and trench perpendicular extension at depths of 100 to 150 kilometer. These observations align with slab tear models that delineate distinct stress regimes, potentially driving ongoing slab segmentation in the northeastern Caribbean. In addition to the profound processes occurring deep within the Earth, the seismotectonic activity of the trench has led to the occurrence of extraordinarily large earthquakes. Peters to the east in the Virgin Islands and Anegada suggest a rupture length of 300 to 4 kilometers and magnitudes reaching approximately 9, highlighting the trench's potential for generating tsunamis across vast basins. Adding to the tsunami risk, multi-beam mapping and ROV surveys have revealed extensive scars from submarine slides and collapse features along the steep carbonate scarp to the north of Puerto Rico. These mass wasting events, which can occur even in the absence of a significant earthquake, have the potential to displace sufficient sediment and water to initiate localized tsunamis. Bathymetric data indicate the presence of large slump heads, such as scars measuring 7 by 3 kilometers, along with retrogressive failures, underscoring the necessity of considering both tectonic ruptures and slope instability in regional hazard assessments. Complex tectonic faulting. The region is situated at a notably oblique plate boundary where the North American plate descends beneath the Caribbean plate at an approximate rate of 20 mm and an azimuth of about 204 degrees. This area also experiences left lateral transform motion. The oblique convergence along a sharply curved margin compels the downgoing slab to bend and wrap around the northeastern corner of the Caribbean, rather than sliding smoothly, as evidenced by bathymetric and tectonic reconstructions of the trench and the nearby insular shelf. Seismic swarms detected by ocean bottom seismographs, OBS, and land stations located north of Puerto Rico indicate the presence of 2 distinct stress regimes, which suggest a propagating slab tear. To the west of 65 degree west, tensile stresses remain trench normal at depths of 50 to 100 kilometers, aligning with typical subduction behavior. Conversely, to the east of 65 degree west, shallow seismic events 50 to 100 kilometers exhibit trench normal tension, while deeper events 100 to 150 kilometers transition to trench parallel tension. This variation in stress orientation with depth can only be explained if the slab has already fractured into 2 segments, each reacting differently to the forces of rollback and bending. On the side of the trench, multi-beam sonar and ROV surveys reveal extensive slope failures along the inclined carbonate platform. Layers that were once laid down near sea level have been tilted northward by approximately 4.5 degrees over the last 3.3 million years, resulting in a steep scarp at a depth of around 4,000 meters. Current imagery displays retrogressive landslide scars measuring up to 7 times 3 kilometers, demonstrating that tectonic flexure not only fractures the slab at depth, but also initiates mass-wasting processes that alter the sea floor. Collectively, these findings, oblique subduction, a slab tear exposed by dual stress fields, and ongoing slope instability, depict the Puerto Rico Trench as an area of remarkably intricate faulting. It is not merely a straightforward subduction trench, it represents a dynamically changing boundary where transform motion, slab fragmentation, bending stresses, and underwater landslides converge, carrying significant implications for regional seismic and tsunami risks. Lysianassid amphipods. Lysianassid amphipods recently discovered in the Puerto Rico Trench, representing a significant finding in our understanding of the deep-sea ecosystem in the Atlantic's deepest area. These tiny, shrimp-like scavengers are part of the family Lysianassidae and are renowned for their capacity to survive in some of the most extreme marine conditions on the planet, including Hadal zones that go beyond 7,000 meters in depth. Their discovery in the Puerto Rico Trench enhances our comprehension of how life adapts to the tremendous pressure, frigid temperatures, and scarce food resources typical of this largely uncharted underwater canyon. Similar to their relatives found in other trenches like the Mariana and Kermadec, Lysianassidae amphipods in the Puerto Rico Trench play a vital role as scavengers, consuming carrion that descends from the upper layers of the ocean. In the nutrient-deficient Hadal depths, they depend on organic material that falls, such as deceased fish, squid, and marine mammals for sustenance. Their uniquely adapted mouthparts enable them to effectively tear into decomposing tissue, which is essential for recycling nutrients back into the deep-sea ecosystem. This feeding strategy supports a varied community of organisms that relies on sporadic food sources in an environment where photosynthesis is not possible. Adapted to withstand pressures exceeding 700 atmospheres and near-freezing temperatures, Lysianassidae amphipods residing in the Puerto Rico Trench exhibit physiological characteristics akin to those found in other deep-sea trenches. Their cell membranes are composed of unique lipids that preserve membrane fluidity under immense pressure, while their enzymes operate efficiently in cold, high-pressure environments. Furthermore, certain species are known to generate protective substances such as aluminum hydroxide gel, which may assist in shielding their bodies from the chemically aggressive conditions present in deep ocean sediments. These adaptations are crucial for their survival in an environment where most life forms cannot thrive. The identification of Lysianassidae amphipods in the Puerto Rico Trench resulted from recent deep-sea exploration missions utilizing remotely operated vehicles, ROVs, and baited traps. These surveys successfully captured footage and gathered specimens from depths of approximately 7,900 meters, confirming that these amphipods are integral to the trench's Hadal community. This discovery is noteworthy as it illustrates that the Puerto Rico Trench, despite its intricate tectonics and extreme conditions, harbors specialized life forms adapted to the depths. It also implies that the trench's ecosystem shares characteristics with other well-researched Hadal environments in the Pacific, enhancing our comprehension of the biodiversity and biogeography of deep ocean trenches. In addition to their ecological significance, Lysianassidae amphipods from the Puerto Rico Trench provide crucial insights into the boundaries of life on Earth. Investigating their biology enables scientists to understand how life persists under extreme pressure and darkness, as well as how organic matter cycles in one of the planet's most isolated habitats. Who knew Sargassum could journey to the Hadal zone? Well, let's find out. Deep-sea isopods. Feeding on Sargassum. Deep-sea isopods, particularly Bathyopsis nibelini, have been spotted consuming floating Sargassum seaweed at astonishing depths ranging from 5,000 to 6,200 meters. This finding, achieved through the use of remotely operated vehicles, ROVs, offers valuable insights into how organic material from the ocean's surface can support life in the abyssal and hadal zones far below. Unlike the majority of deep-sea scavengers that depend on decaying carcasses or marine snow, Bathyopsis nibelini showcases a unique feeding strategy focused on the consumption of these brown algae fragments, highlighting an intriguing connection between surface ecosystems and the deep ocean floor. Bathyopsis nibelini is equipped with serrated mouthparts that are specifically adapted to grasp and process the tough, fibrous Sargassum fronds. These specialized mouthparts allow the isopods to effectively bite and shred the seaweed, which would otherwise be challenging to digest. Footage captured by ROVs has documented these isopods transporting pieces of Sargassum, indicating active foraging behavior instead of mere passive scavenging. This behavior contradicts earlier beliefs that deep-sea isopods are restricted to feeding on carrion and uncovers a broader dietary adaptability in these extreme habitats. Recent laboratory studies on Bathyopsaurus nibelini have revealed that these isopods possess a unique gut microbiome that can effectively break down the complex polysaccharides found in Sargassum. The microbial communities within them generate enzymes that dismantle algal cell walls, enabling the isopods to absorb nutrients from materials that are typically challenging for most deep-sea creatures to process. This symbiotic interaction illustrates the evolutionary adaptations essential for thriving in nutrient-scarce, high-pressure environments where energy resources are limited. Moreover, the feeding behavior of Bathyopsaurus nibelini on Sargassum highlights the ecological significance of Sargassum mats that drift across the ocean's surface. While these mats are frequently examined for their effects on coastal ecosystems, they also serve as biological subsidies, ferrying organic matter from the sunlit surface waters down to the deep ocean. As fragments of Sargassum sink or break apart, they offer a rare source of carbon and energy for deep-sea benthic communities, including isopods, amphipods, and various other invertebrates. This mechanism plays a crucial role in sustaining life in areas that are far removed from the primary productivity associated with sunlight-driven ecosystems. The revelation of Bathyopsaurus nibelini's feeding behaviors through ROV missions carries significant consequences for our comprehension of deep-sea food webs, particularly within the abyssal plains and trenches of the Atlantic Ocean. This finding highlights the manner in which energy transfer between vastly different oceanic layers can sustain intricate life forms that have adapted to extreme environments. Additionally, the isopods' capacity to utilize Sargassum could affect the responses of deep-sea ecosystems to variations in surface productivity driven by climate change or altering ocean currents. This finding uncovers a distinctive ecological adaptation that includes morphological, microbial, and behavioral characteristics, allowing survival in one of the most demanding habitats on Earth. Not only does this discovery enhance our comprehension of deep ocean biodiversity, but it also highlights the connection between ocean surface and deep environments via the movement of organic matter like Sargassum. In darkness and cold, there are vents that glow with biological activity, unlike any other. Hydrothermal vents. In 2022, the enhanced deep-sea submersible Alvin achieved remarkable dives to about 6,400 meters along the slopes of the Puerto Rico Trench, where researchers recorded the first-ever hydrothermal vent systems in this section of the Atlantic Ocean. These hydrothermal vents signify an extraordinary geological and biological occurrence, where mineral-rich fluids rise from the ocean floor due to intense tectonic and volcanic activity beneath the Earth's crust. While these vents were not located in the trench's deepest Hadal zone, 8,400 meters, their discovery on the trench slopes underscores the dynamic and intricate nature of the region's underwater geology and ecosystems. Hydrothermal vents develop when seawater infiltrates fissures in the ocean crust, is heated by underlying magma or hot rock, and subsequently ascends back through the sea floor, transporting dissolved minerals and chemicals. When these mineral-rich fluids encounter the frigid deep ocean water, they precipitate to create chimney-like formations and deposits around the vent openings. The vents identified at a depth of 6,400 meters in the Puerto Rico Trench area release hot, chemically abundant fluids that sustain unique biological communities not found anywhere else on the planet. One of the most fascinating features of hydrothermal vent ecosystems is their dependence on chemosynthesis instead of photosynthesis. In the dark depths where sunlight is absent, vent microbes utilize chemicals like hydrogen sulfide, methane, and metal ions from the vent fluids to create organic matter. This chemosynthetic mechanism establishes the foundation of a complex food web that sustains a variety of organisms, including specialized bacteria, tube worms, crustaceans, mollusks, and other invertebrates. The identification of these vent systems on the slopes of the Puerto Rico Trench thus paves the way for new research into life's ability to adapt to extreme environmental conditions, broadening the known habitats of vent communities beyond the extensively studied sites in the Pacific Ocean. Geologically, the existence of hydrothermal vents along the trench slopes is associated with the region's active tectonic activity. The Puerto Rico Trench represents a complex plate boundary where the North American and Caribbean plates interact, leading to faulting, crustal deformation, and magmatic activity that serve as the heat source fueling hydrothermal circulation. The vents that have been discovered are linked to exposed intrusive oceanic crust, estimated to be as much as 100 million years old, suggesting a long and evolving geological history. Furthermore, the active fault lines and submarine landslides observed during the Alvin dives provide further evidence of the ongoing dynamic processes that shape the sea floor and influence the formation of vents. In addition to their scientific importance, these hydrothermal vents attract attention due to their mineral resources, frequently containing precious metals like copper, zinc, and gold. Gaining knowledge about vent systems in the Atlantic Ocean is also crucial for evaluating the potential environmental consequences of deep-sea mining operations, which is becoming an increasing concern as technology progresses. Moreover, exploring these ecosystems provides valuable information about the conditions of early Earth and the likelihood of comparable life-sustaining environments existing on other oceanic bodies such as Europa, a moon of Jupiter, or Enceladus, a moon of Saturn. They emphasize the geological intricacies of the trench and uncover a vibrant ecosystem that relies on chemosynthesis, flourishing in one of the Earth's most extreme and least studied environments. High-pressure labs have cultured bacteria that laugh at 600 bar of crushing force. Deep microbial ecosystem. The Hadal zone of the Puerto Rico Trench, which descends beyond 6,000 meters to depths that surpass 8,200 meters, is home to microbial ecosystems that challenge our comprehension of life's boundaries. Despite these trenches, covering less than 1% of the ocean floor, their microbial communities exhibit distinct genetic and metabolic characteristics shaped by extreme pressure, low temperatures -2 degrees Celsius, and constant darkness. Single-cell genomics conducted on 4 organisms collected at a depth of 8,219 meters identified members of Thaumarchaeota, SAR11, and 2 proteobacterial lineages associated with known piezophiles. Comparative studies with shallow water relatives revealed trench-specific genes, mixotrophy pathways in Thaumarchaeota, such as glycine cleavage, a complete glycolytic suite in SAR11, and hydrogen or nitrous oxide oxidation in Cycromonas-related cells, demonstrating unique adaptations to life in the Hadal zone. Metagenomic sequencing of the waters in the Puerto Rico Trench, amounting to approximately 145 MB of unique data, illustrates a level of genomic complexity that surpasses that of surface oceans. The average estimated genome size of trench microbes, -3.57 MB, significantly exceeds that of surface assemblages, -1.75 to 1.85 MB, indicating a broader genetic toolkit to manage chemically diverse and resistant organic matter found at such depths. Genes involved in signal transduction, especially those related to redox and oxygen sensing, are notably overrepresented, along with sulfatases for breaking down complex polysaccharides, heavy metal efflux systems, and a variety of transporters designed to scavenge limited nutrients. Beyond genomics, the trench has revealed culturable piezophiles. In 2008, seawater collected from a depth of 6,000 meters aboard the RV Atlantic Explorer led to the discovery of the novel isolate Profundi Monas Piezophila. From an ecological perspective, these profound microbial communities are essential in driving biogeochemical cycles within the Hadal zone. By oxidizing methane and hydrogen, replenishing nitrogen and sulfur reserves, and recycling sinking organic matter, they support higher trophic levels, ranging from amphipods to holothurians. Their diverse genomes and unique enzymes present significant biotechnological opportunities for bioremediation and biocatalysis in extreme environments. Additionally, the microbes found in the trench act as analogues for possible extraterrestrial life, providing valuable insights into the limits of life on icy, oceanic worlds. Sediment geochemistry and carbon flux. Core sediment cores extracted from depths greater than 8,000 meters in the Puerto Rico Trench unveil a stratigraphy primarily composed of pelagic clay, interspersed with episodic turbidite layers that signify intense mass-wasting events, channeling continental shelf debris into the Hadal zone. These fine-grained hemipelagic clays accumulate gradually in the perpetual twilight of the deep ocean, layered with graded sand to silt deposits formed by turbidity currents instigated by storms, underwater landslides, or seismic activity along the trench's steep inclines. Geochemical evaluations of these cores reveal total organic carbon TOC levels ranging from 0.2 to 0.5%, with C-N ratios hovering around 6, suggesting a primarily marine phytodetritus origin rather than terrestrial sources, aligning with earlier USGS findings from the trench. Despite an overall carbon flux below 1, one of the lowest in the global ocean, these intermittent turbidity occurrences serve as effective transporters, channeling reactive, labile organic matter straight into the Hadal environment before it can be oxidized in midwater or sequestered on the continental slope. Once buried beneath subsequent clay layers, this fresher carbon becomes accessible for microbial mineralization through sulfate reduction, denitrification, and fermentation. Maintaining unexpectedly high rates of benthic oxygen consumption akin to those found in much shallower, more productive ecosystems. The thickness and frequency of turbidite beds influence how effectively organic carbon is buried. Thicker, coarser layers shield trapped detritus from oxygen-driven degradation, facilitating deeper burial and subsequent use by subsurface microbes in areas of sulfate, methane transition, and methanogenesis. At the same time, the pelagic clay in between, lacking fresh organic matter, serves as a barrier, restricting upward diffusion and maintaining pockets of labile carbon. This alternating sedimentation pattern also leaves its mark on the genetic makeup of trench microbial communities. Metagenomic analyses of over 8,200 meters of water samples show a significant abundance of sulfatase genes, enzymes that break down polysaccharides, and pathways for alternative electron acceptance such as nitrous oxide and hydrogen oxidation, which are adaptations believed to enhance the use of the sporadically delivered organic materials. Essentially, the Puerto Rico Trench functions like a V-shaped funnel. Its structure and tectonically induced mass wasting introduce fresh carbon to Hadal depths, where layers of pelagic clay then isolate and preserve it, resulting in a mosaic of reactive substrates that supports a complex, detritus-driven food web. Although the average carbon flux is low, the increased burial efficiency provided by turbidites and the trench's distinctive topography amplify the effects of each input event, creating hotspots of microbial activity and sustaining the trench's renowned alien-like fauna. Aged North Atlantic deep water carries a trace of Antarctic influence into the trench. Bottom water properties. The Hadal waters of the Puerto Rico Trench, sampled directly from depths approaching 8,300 meters, display physical and chemical traits that not only reflect their origins in North Atlantic deep water NADW, but also highlight the distinct alterations brought about by Hadal isolation. In 2022, the University of Puerto Rico conducted free vehicle deployments fitted with high-precision CTD profilers capable of reaching depths of 9,000 meters, which recorded a nearly stable temperature of 2.7 degrees Celsius at the trench floor. This temperature is significantly warmer than the sub-zero bottom waters found in Pacific trenches, yet it aligns with the characteristics of NADW that has aged and mixed during its journey from deep water formation sites. At the same time, salinity readings remained around 34.6 PSU, which is consistent with the salinities typical of NADW, 34.0 PSU diluted by a minor fraction, approximately 18% of Antarctic bottom water AABW. This mixing ratio has been inferred from radiocarbon and tracer studies carried out by the Deep Autonomous Profiler, DAP, during sea trials to depths of 8,377 meters. The properties of these bottom waters position the trench waters just outside the pure NADW range on temperature salinity diagrams, indicating slight influences from AABW while still retaining the core signature of aged Atlantic deep water. One of the most notable aspects is the remarkably low concentration of dissolved oxygen, which is recorded at around 4 in analyses of benthic chambers and water samples. This figure ranks among the lowest documented worldwide outside of permanently stratified basins, significantly influencing the microbial ecology of the trench by limiting aerobic respiration to the upper millimeters of sediment and promoting the presence of facultative anaerobes and microaerophiles in pore waters. The depletion of oxygen is attributed to a combination of slow bottom water renewal, estimated turnover times of approximately 300 years, and relatively high benthic oxygen consumption driven by sporadic organic pulses delivered by sediment. In situ, incubations within benthic chambers in similar Hadal environments have reported oxygen consumption rates reaching up to 600, suggesting that despite the low average oxygen levels, benthic communities are actively respiring and recycling organic carbon. Furthermore, the interaction of temperature, salinity, and oxygen plays a crucial role in the density stratification that separates trench waters from the deeper waters above. The slight increase in density from the colder, saltier Antarctic bottom water, ABBU, injections creates a stable layering that restricts vertical mixing, allowing for the maintenance of fine-scale biogeochemical gradients. These gradients subsequently affect nutrient transport, microbial zonation, and the distribution of megafauna, many of which depend on the thin oxygenated layers at the bottom for respiration or anaerobic metabolic processes within sediment interstices. Notably, the nearly constant temperature of 2.7 degrees Celsius establishes metabolic baselines for enzymes adapted to Hadal conditions, while the stable salinity maintains osmotic balance for piezophiles that are sensitive to variations in ionic strength. If you enjoyed this video, like and subscribe, and also click the next video shown on your screen.